All right, well, I'll get started because I think I've probably put too many slides in this presentation, so I'll make sure that I can get through them. So I was thinking about something to present for the webinar, and there was a few things to draw from, I suppose, from my research career, and indeed, one idea was to sort of title this talk about my career in torturing plants, which you'll get a flavour of, sense of, sort of through this talk. But I thought it was a little bit macabre. And indeed, I do grow a lot of plants that grow into nice, healthy trees and vegetables as well. So I ended up landing on this paper, which wasn't my own sort of efforts here. This came from Jazz, who shares the office here at UTAS with us, and it kind of dawned on me that it would be a good opportunity to talk about my experience running drought experiments, sort of under control conditions in particular, partly because the centre is very much interested in drought and how plants are going to respond to drought, but also sort of it occurred to me that a lot of those experiments were very much genetics focused, and that it would be useful for people running genetics type experiments to get a sense of how it is that you might sort of understand the physiological response of plants in response to drought. So this paper, Junger and Versluis, I'm not sure, I actually don't know those guys, but it's their focus on the paper was really about understanding the importance of measuring the plant water status during drought experiments. So I'll basically so as a presentation outline, I'll sort of take a step back and talk a little bit about some of my research plants in the field and how that might relate to the centre, and then go through sort of a couple of different approaches that you might take when studying drought and the response of plants, and then sort of hopefully sort of spend most of the time focusing on the glasshouse experiments and asking questions about when and how and what to measure, indeed what how to measure your plant water status and overcoming issues like plant size effects. So these slides generally sort of a feature in drought related talks, and of course I won't spend too long on it, but drought obviously is a major environmental stress, and indeed it's a major evolutionary driver that shaped species distributions and indeed what species look like. The concern is that drought is becoming more severe and intense, primarily as a result of global heating. So as you get warmer temperatures, that variable rainfall that occurs naturally, you end up getting more evaporation from the soil and greater water loss from the plant. And so that higher VPD, essentially that's what it translates into, creates more physiological stress for those plants. And you end up getting more incidences of drought mortality, like you see globally, sort of on that caption on the right, but also like we saw in New South Wales in 2019. Of course, drought also affects crop production, and indeed, I don't have to tell you guys that. And the caption on the right is essentially just coming from the IPCC report in 2022. Showing that the incidence of, or the chance of incurring sort of a drought, an extreme drought in any particular year is increasing substantially with global warming. And through the latest El Nino report from the BOM in Australia, just to sort of emphasise that we've had 3 years of rain, sort of a La Nina system, in Australia dominating sort of climate here. Which has been associated with lots of wet weather, and so people tend to forget about that. It does dry out, and that drought is sort of a feature of our climate in Australia. Farmers certainly don't forget it so easily, I suspect. But we are coming into an El Nino weather pattern again, which suggests that the southeastern Australia in particular might have a greater chance of seeing dry and warmer weather. It's not all gloom and doom, of course. As researchers, we're in the business of trying to understand, how plants are adapted to drought, and also to improve through experiments, which I'll get onto in a minute, I promise, in terms of managing natural systems and improving productivity of crops. And here's a bunch of papers that basically have attempted to do this. So let me just take a step back and talk about aridity gradients. And I kind of wanted to throw this in just because it kind of links in with our work in the youth project and how that might tie in, to the centre's goals of integrating knowledge from nature into agricultural systems. And I think I've sort of said it in the talk before, probably a long time ago, that when you sort of study, plant distributions and the traits of plants across aridity gradients, you're essentially kind of examining the outcome of sort of nature's largest and longest ongoing drought experiment. And it's just how, and in the sense that, excuse me, the traits that you see across that aridity gradient have been shaped by evolution and the impact of, and as a result of those differences in precipitation and how that affects drought stress. And so we see species that are adapted to those different climates, for example, in this case, with species of eucalypt showing increased vein density, and another example of where species from arid environments have smaller, narrower vessels. And so here's a photo of me and Sean Gleeson working in central New South Wales. If Ian Wright is watching, he might recognise Round Hill as one of the sites that he's used in his work at Macquarie University. And I put that up because fieldwork is fun. You sort of gets you out into the field, but it can be logistically challenging. And I think maybe that's sort of a point of contrast with I suppose the control and sort of the convenience that you get sort of from running glasshouse experiments. So aridity gradients are also useful for studying within species variation for those species that occur across sort of aridity gradients, and that way you can start to sort of examine genetic differences between populations. Long-term monitoring sites are super useful. These are some images of a site in Switzerland. The Swiss canopy site. Ernst Carmen presented some of this data a little while ago. Some of you might have seen, but essentially they've been monitoring sort of the plant water status in these trees where they've set up a crane to access the canopy, and shown that, and by chance, have sort of encountered a very strong drought event. So they were able to actually monitor that water potential and really show how the trees were responding to drought in real time. In Australia, we do have some long-term monitoring plots, and not many of these are sort of administered by the Turn kind of research infrastructure. And it's certainly useful, but it tends to be a little bit, in terms of sort of some of the drought response traits that people are measuring, it tends to be a little bit piecemeal. I think it's fair to say that the drought that we were witnessing in New South Wales, that Rachel Nolan and Brendan Choate were very much focused on in 2019, we were still tracing the drought. We didn't have a long-term monitoring plot set up where we were able to capture that sort of change in plant water status and the stress. We're essentially just chasing where the drought was most heavily impacting trees at the time. Another approach is to exclude rainfall from the system itself. So there's a little bit of control being, sort of applied here. Of course, it's very expensive, and but super interesting experiment to set up. This is in the Daintree, and you can see that it's been there for several years now. I'm not exactly sure how long, but I think this data is from 4 years of research, and the rainfall exclusion takes water off the site, and you can see that there's sort of an increase. Sorry, sort of a decrease in the water content in the upper layers of soil in particular. And what these researchers are reporting is that trees are responding to that decreased surface water in particular by trees developing deeper roots. So that's certainly an interesting result in showing just how trees are responding to an experimental drought. Slightly more controlled is we have sort of rain exclusion experiments with a fairly significant setup still. So on the left is a rain exclusion site, a shelter at the HIE or Western Sydney, where basically sort of the roof and the walls sort of can be opened or closed, depending, like if it's closed, if there's sort of a rain event is approaching. And in that way, you get to grow bigger trees, either in the soil or in big pots. And so you get to sort of do experiments on larger trees. And this is also true for a polytunnel type setup. And on the right is another experiment where we had, I think just in the background, you can see sort of a rainfall exclusion canopy that was would move across the trees as soon as rainfall was approaching. So let's get down to the glasshouse experiments. And of course, there's many benefits. Essentially, you get to control the level of the drought. You can't so much with all these other approaches. You can control the temperature and the humidity and the day length. You run experiments for a short period of time or for longer, and can study in greater detail, maybe with access to sort of more, sort of expensive equipment or physiological tooling, all the different sort of responses that you might be interested in. And with that level of control, you get to sort of identify mechanisms of drought tolerance to a greater level than some of those other experiments, and indeed identify phenotypes that may be tolerant to drought, that may be interested in moving forward in plant breeding, etc. So there are the benefits, but of course there's plenty of risks. And so here's a couple of slides that some of you are probably familiar with. The one on the left is the impact of a major hailstorm event at ANU, where they've basically destroyed all their glasshouses. It was super, dramatic and sad. I think it's still sort of being repaired, so it's sort of a major sort of event for those research projects. And indeed, the flooding in Queensland has caused major events, major disruption to those experiments at Queensland University and other universities too, I presume. Indeed, I've not had that sort of impact on experiments that I've run, but certainly when the air conditioner fails in a glasshouse in Western Sydney, or even any sort of sunny day, you really need to get your plants out of those glasshouse cells, because temperatures can easily sort of exceed 50 degrees without even trying. So the types of drought experiments that you might run in a glasshouse include a sort of a soil drought experiment, and that's essentially where you're sort of applying a treatment and controlling for the level of soil water deficit that the plants are exposed to. So in a way, yeah, you're sort of controlling for the soil water content and letting the response of the plants inform, sort of how their sort of rank and drought tolerance, or when, the type of response that you see. Controlled irrigation is ideal for this. I've run experiments weighing pots, basically trying to sort of homogenize the level of drought stress across plants, different size. We'll get onto that in a minute, and trying to sort of track, sort of manage that drought experiment based on sort of the water use of the sort of the smallest plant. So you're sort of re-watering sort of larger plants, to a greater extent than others, and it's an absolute nightmare. So controlled irrigation is definitely the key. Another sort of form of drought might be just a physiological drought you might be interested in. And for these, you drive plants down to a common level of water potential, which is a little bit similar to the soil drought approach above. Or you may sort of dry plants down to a common level of physiological stress. So these are some data from Tim and Hervé, where they've dried down plants in a controlled experiment to a level of water stress associated with a particular level of hydraulic dysfunction. It's sort of associated with a 50% loss of hydraulic function in the stems. And essentially been able to determine that that level of hydraulic dysfunction was associated with the sort of drought survivorship. It was that level of drought stress that was connected with whether or not a plant would recover or not. So we're able to sort of say that this level of hydraulic dysfunction was connected with mortality in trees. And the final approach is sort of just a dry down experiment. We just let plants dry down, sort of follow it from a well hydrated state. And indeed, there's issues with plant size when we get to that. Glasshouse experiments tend to sort of be focused on seedlings. Things to consider might be the temperature that you dry your plants down under. So of course, a cool temperature regime will allow for a slower dry down. It's easier to manage. I've had experiments where it's just been a little bit too hot, or plants are a little bit too big relative to their pot size, which is another point there. And the plants just dry down too quickly. So you miss the chance to actually sort of assess the physiological function that you're interested in. And it's not particularly realistic either. And of course, there's a bunch of interactions you can have with your experiments, including different CO2 treatments and temperature and nutrients. The fact is multifactorial sort of designs can quickly get out of control. So certainly I think, that's wise to keep things fairly simple, so it maximizes your chances of identifying a specific response to those treatments and for identifying mechanisms. The duration is another sort of a consideration. Short-term experiments are fine for just sort of examining sort of initial responses, but you might be interested in how plants adjust plastically. Let's go over a long-term drought experiment where plants are growing under different levels of water deficit for long periods of time. And indeed, you may want to rewater your plants to look at how plants recover following drought. And then again, there's another approach of just drying down plants, to death to sort of determine sort of survivorship. What to measure? I'll go through this super quick because you don't need to read any of all those traits, but essentially you can measure anything. Depends on what you're interested in measuring. So what I've listed there from Brendan's paper is a bunch of physiological and hydraulic traits, mostly about how plants, sort of the type of traits that might influence how plants respond to drought. But if you're interested in hormones and genes and their interactions, you might be interested in different stages of drought stress. So if I quickly just go back to this paper that Jazz sent through to me, and why it's important for people to understand plant physiology during drought experiments and to understand the plant water status. And I think this graph sort of sums it up. Basically, the importance of measuring plant water potential, or the level of your plant water status, is the plant water potential that responds to the soil dehydration during drought. So you can see that if a drought starts at day 0, and over time the soil dehydrates and water potential starts to decrease in the soil as well as in the plant. It's sort of just a fundamental physical response of that tension within the plant decreasing during drought. There's a whole bunch of physics in that, which I won't go into in terms of how plants move water, but the key thing is that water potential is fundamentally decreases during drought. It's a measure of the plant water status. And what else? And sort of having that, so if you dry a plant down, you tend to get sort of a response from in the stomata, which they close at a certain point, at a certain water potential, right? And then you get a sort of other processes where you start to get that drought stress occurring, which might be associated with loss of hydraulic function, associated with 50% loss of hydraulic function, and then 88% loss of hydraulic function, which is close to that mortality threshold. And once you know the water potential of these different thresholds of drought stress, you can kind of compare across species and start to sort of understand the coordination between traits, sort of linked to this response to water potential, and this ranking as well, like based on the species tolerances. So measuring plant water status, typically we use the pressure chamber. So here's a picture of a pressure chamber from PMS. You can buy those from that company. But essentially it applies pressure to a leaf that you excise from the plant at different level during that drought experiment. You put it inside the chamber with the PDL sticking out of the top, and then you apply positive pressure to that plant. And then once that positive pressure equals the tension that's sort of inherent in the leaf at the time of that excision, it starts to exude water out of the cut end, and then that's your water potential that you jot down. Other methods, so I should say that that this chamber is super useful, and I mean you've got to be really careful in its use, but you can do lots of quick measurements across different plants. Soft leaf species can be a little bit challenging because they get a bit squashed in the casket, but otherwise you can quickly scale up lots of different measurements across different treatments or different individuals under different treatments. So useful in that sense. Cyclometers are useful for giving you continuous data of a plant response during drought. Disadvantage is that that you sort of tend to only be able to set up one sort of per plant, and with limited numbers, you know, because of the expense and I suppose sort of the work and effort in getting them going. But it could be, if it's a representative plant, you know, you could sort of use it as a surrogate for the response of the other plants. And indeed, I need to mention, of course, the dendrocams developed here with Tim and Chris Lacani. And they give you real-time measurements of water potential of the plant during drought. And so this is just an output. This is what you see inside the camera, and the expansion and contraction as the plant transpires during the day and starts to decrease as drought stress increases. You see this sort of signal shown in the graph. So once you are sort of, I suppose, convinced that you need to measure water potential, you know, you might sort of start to think about sort of other traits that are of interest during the drought. And I've just listed a few of these, you know, and especially if it's interested in the genes that are controlling them, the hormones that interact with this sort of response. And so during early stage of drought, this is sort of a layout of the sequence of these events. And what you tend to get is the stomata will close as drought stress increases, the leaves will wilt, that's sort of a turgor response. And xylem cavitation starts to occur in the water transport system. Toward the end of drought stress, you know, as plants becoming particularly drought stressed, you get sort of the photosynthetic systems, which is measured as a FV on FM response. Some of you might be familiar with. That tends to crash just before the leaves, you know, completely desiccate and die. So there's a bunch of sort of physiological responses here that, if you're interested in measuring other aspects of the hormones and genes, that you could sort of relate to how plants are responding physiologically in response to changes in water potential. I won't go too much about these, but these are just some of the tools that you can use to measure some of those traits. And so just to talk about plant size just quickly before I finish, we have this this diagram that's from the paper that from these two characters who emphasize the need for measuring plant water status. And they kind of talk about these different ascensions of Arabidopsis or whatever plant you're interested in, where one is smaller and one is larger, and then you apply the same drought treatment to each. And essentially, because the smaller plant uses less water in that early stage of drought, while there's still some access to water in the soil, it's going to dry down more slowly than a plant that's bigger, just with the fact that it's got less leaf area, less demand for water use. And so if you're not accounting for plant size, you're going to get a very different response. And indeed, you might sort of assign that smaller plant, which has used less water and therefore become has been drought stressed as a larger plant, which has used up all its water and started to basically desiccate. You may assign greater drought tolerance to this smaller plant, which actually would be not true. You can measure the differences in plant size before you start your experiment. This is an experiment with Globulus from UTAS, PhD student, Hans. And he's measured the plant size before the drought experiment and factored that into the model by, in assessing drought tolerance in terms of survivorship. So some have died, some stayed alive, and many of those that have stayed alive will probably be smaller than some of the larger ones. But you can start to sort of factor in a plant size effect with, as long as you've measured it, in statistically in the model. Otherwise, we go back to that controlled irrigation setup where a larger plant essentially can be sort of comparable with a smaller one if you can apply a little bit of extra water to it to control the level of soil water deficit. So it's comparable with the plant that's smaller and using less water. And finally, I'll just talk about some of the experiments that I've been doing recently, where we're able to control for plant size effects by growing plants together. And the idea is that with plants that are growing together, having access to the same soil water, even a larger plant that's using up more water than a smaller plant in the early phase of drought, because the roots are intermingled throughout that soil, it's drawing down water for all the plants, not just for itself. So it means that all plants are going to be experiencing the same level of water deficit. The water potential should be homogeneous as those plants dry down during the early phase, at least. So I'll press play, that's working. This is the response. It should only take about 20 seconds, I think, of just trays of 10 I think there's 20 plants in that tray, 10 species, so two reps per species, and they're drying down over the course of 10 days. And I'll be measuring the health of the photosynthetic systems on the right. And there's some clear differentiation in the response of those different species of eucalyptus, which is very nice. And importantly, the water potential, and it's not super clear from this graph, but up to sort of day 5, the water potential was fairly homogeneous among those different plants. And indeed, the variation that you do see in the water potential didn't determine the sort of the final outcome in terms of that drought survivorship. So that was encouraging. After day 6, the water potential start to separate because of the different levels, the different sort of tolerances of those different species. And as the plants really sort of die and crisp up, then the water potential crashes dramatically. So that's it. So rather than sort of going on for too much longer, I'll just sort of leave it at that, and yeah, let's open up to questions if people have them.